There's a plant fiber your body can't digest at all. It passes through you whole. And in a new study, adding it to a diet loaded with sugar didn't just slow liver damage down. It reversed damage that had already happened, while the sugar kept coming. Every sugary drink you swallow is really two sugars fused together, glucose and fructose. Your body handles them completely differently. Glucose gets used almost everywhere. Fructose gets funneled almost entirely to one organ, the liver, where in excess, it gets converted straight into fat. Fructose consumption has climbed roughly 100-fold over the last century, mostly through high-fructose corn syrup, and your liver hasn't caught up to that shift. Scientists at the University of California, Irvine wanted to know if there was a way to change fructose's route before it does damage, not by cutting sugar out, but by changing what travels alongside it. Their subject, a fiber called inulin, found naturally in garlic, onions, and asparagus. Chemically, it's just one glucose molecule attached to a long chain of fructose molecules strung together. But human digestive enzymes can't cut that chain apart, so it passes through completely intact. Their test subjects were male mice, not people, and that distinction matters for everything that follows. Here's why fructose gets special treatment in the first place. Unlike glucose, which cells across the body compete to use for energy, fructose is metabolized almost exclusively by the liver, and it's a much more aggressive trigger for fat production there, switching on the genetic machinery for building new fat molecules far more forcefully than glucose does. Breaking fructose down inside liver cells also produces a reactive byproduct capable of damaging DNA, RNA, and proteins, adding a slow drip of oxidative stress with every exposure. There's a second route to the damage that has nothing to do with the liver directly. Gut bacteria that encounter unabsorbed fructose can convert it into a small molecule called acetate, which travels to the liver and gets used as raw material for building even more fat, meaning some of the fat clogging the liver never even passed through the liver as sugar first. It arrived pre-converted, courtesy of bacteria the person never asked for help from. Excess fructose has also been linked to a leakier, more permeable gut lining, longer intestinal villi, and shifts in the gut's bacterial population that aren't especially healthy. The kind of low-grade dysbiosis that shows up long before any blood test would flag a problem. Stack all of that up over weeks and months, and you get hepatic steatosis, fatty liver disease, creeping in through several doors at once, not just one. To test whether inulin could shut those doors, the team, led by Chol Soon Jang, ran mice through a controlled feeding experiment. One group got a standard diet. Another got high-fructose corn syrup for 16 weeks, long enough to firmly establish fatty liver. A third group got the high-fructose diet alongside inulin from the very start. And in the most demanding test of all, a fourth group ate the sugar-heavy diet for the full 16 weeks first, developing fatty liver exactly like the second group, and only then had inulin added on top for another 14 weeks, sugar and all. To track exactly where the fructose molecules were actually going, the researchers used carbon-13 labeling, attaching a heavier, trackable version of carbon to fructose molecules before feeding them to the mice, then following those specific atoms with mass spectrometry to see how much ended up as fat in the liver versus how much got broken down before it arrived. The results: mice getting inulin alongside fructose gained less weight and carried less liver fat than the sugar-only group. And the fourth group, the one already carrying existing liver damage, saw that damage measurably regress, along with a drop in liver fibrosis markers, even though the sugar in their diet never stopped. A closer look at the actual fat molecules inside their livers showed drops across several specific harmful lipid types, including the same ceramides and triacylglycerols that show up in human fatty liver biopsies. So is inulin just working because it's less caloric or because it's fiber in general? The researchers checked both. Swapping inulin in for cornstarch did shave off a small amount of calories, but total calorie intake across the sugar-fed groups came out statistically indistinguishable. This wasn't a story about eating less. To confirm the gut microbiome itself was doing the work, rather than inulin acting some other way, the team took the adapted bacterial communities from inulin-fed mice and transplanted them into new mice that had never eaten inulin at all. The protective effect came along with the transplant. That's about as strong a piece of evidence as microbiome research can offer. Not just a correlation between fiber and better liver numbers, but bacteria that carry the effect with them into an entirely new body. Digging into which microbes mattered most, one species stood out. Bacteroides acidifaciens, an ordinary gut resident that most people carry without ever hearing its name, which, once the mice were eating inulin regularly, appeared to specialize in breaking fructose down before it could travel any further. This wasn't a single hero bacterium acting alone. The effect involved a broader shift across the gut community. But Bacteroides acidifaciens kept showing up as a key operator whenever the researchers looked closely at the data. Here's the detail that overturns the assumption most people carry about gut bacteria. When you hear gut microbiome, you probably picture the colon. That's where the overwhelming majority of gut bacteria live, by a huge margin, and where most microbiome research points its cameras. But when the researchers traced exactly where the fructose interception was happening, it wasn't primarily in the colon. It was happening earlier, in the small intestine, a stretch of gut usually treated as too fast-moving and too oxygen-rich to host much bacterial activity, and largely overlooked by microbiome science for exactly that reason. Inulin was reshaping that normally quiet stretch into the actual front line of defense, catching fructose before it ever reached the liver, and before most of it ever reached the colon at all. Inulin also had a second trick, independent of bacteria entirely. It switched on the liver's own internal antioxidant factory, boosting production of glutathione, one of the body's core defenses against exactly the kind of cellular damage fructose causes. Two separate defense systems, triggered by one plant fiber. None of this has been tested in people yet, and mice aren't a guarantee of how a human liver responds. But the foods naturally rich in this exact fiber are already familiar: garlic, onions, asparagus, bananas, leafy greens. That's not a prescription. It's just worth knowing that a fiber your body can't even digest isn't inert cargo passing through you. It's raw material for a bacterial community that, in this study, learned to intercept sugar overload from the inside, catching it before the liver ever saw it. High-fructose corn syrup isn't going anywhere anytime soon. It's cheap, and it's in everything from bread to salad dressing. But if this pattern holds up in human trials, one of the more promising counterweights to that might already be sitting in the produce aisle, not a pharmacy.